Bueno, buenos días. Bueno, me vais a permitir que presente a Bruno en castellano, aunque va a dar la charla en inglés. Bueno, es investigador en el Departamento de Biología del Cáncer del IIB, del Instituto de Investigaciones Biomédicas de Alberto Sols, pero además es parte del IRYCIS, no solo parte del IRYCIS, sino que es el codirector del área 3. Y realizó su tesis doctoral en la Universidad de Tulane en Nueva Orleans y tuvimos la suerte de que en el 2011 decidió venirse a España, al CNIO, y ya empezó a trabajar ahí con cáncer de páncreas y con células madres del cáncer. Actualmente es el líder del grupo BioPac del área 3 del IRYCIS, además de ser el codirector. Y bueno, creo que nos va a dar un seminario muy interesante. Muchas gracias, Bruno, por aceptar nuestra invitación.
Gracias. I'm going to present today in English, if that's okay with everyone. And I'll try to be quick because we have another jornada for the postdoctoral students right afterwards. Okay, so what I would like to do today, if I can achieve it, is to give you sort of a glimpse, snapshots of what we do in the laboratory. Mainly, our focus is on cancer stem cells in pancreatic cancer, although we do touch other types of cancers as well. So I'll show you some data, for example, with colorectal cancer. Okay, so I think it's important when we initiate a talk about cancer to put it in context, specifically pancreatic cancer and other cancers, as far as are we losing the war against cancer? And I think one of the best reflections is 5-year survival rates. So when we look at some types of cancer, for example, breast and prostate, we're actually making great accomplishments in improving overall patient survival. This may be due in large part to surveillance programs that we have to detect breast cancer early and to detect prostate cancer early. But if we look at other types of cancers, for example, here, pancreatic cancer, lung cancer, liver cancer, we're definitely not meeting our expectations. We still have a 12% 5-year overall survival for patients, which is not where we want to be. And if we look at the history, for example, this is from 1960 and projections all the way to 2030, we have seen a 17% drop in mortality, which is fantastic. But if we look at two particular types of cancers, lung cancer and pancreatic cancer, we see that pancreatic cancer and lung cancer the mortalities are increasing, and pancreatic cancer is expected to become the second leading cause of cancer in the United States by 2030. So these are not good statistics for pancreatic cancer. So briefly, pancreatic cancer is a malignant neoplasm of the extracrine pancreas. It is the 4th most frequent cause of cancer-related death, deadliest form of cancer, one of the deadliest forms of cancer, 5-year survival rate of anywhere from 12 to 13%. That depends on the continent and the country. And this is primarily due to these very horrible statistics, the lack of early symptoms and reliable diagnostic markers to detect pancreatic cancer early. So unfortunately, PDAC is usually diagnosed at very late stages when the tumor has already advanced to unresectable metastatic stages, as you can see here, metastasis in the liver, and this is obviously a macro image of a liver with pancreatic tumors or metastases. So pancreatic, if this wasn't bad enough, pancreatic tumors are highly resistant to chemotherapy, also to radiotherapy, and therapeutic options do not cure PDAC. We can offer patients with advanced disease prolonged of life anywhere from 6 to 12 months, but this isn't the best case scenario. And in fact, as well, I found this morning, the National Cancer Institute Surveillance Epidemiology and Results Program estimates that in the United States, 66,440 people will be diagnosed with pancreatic cancer in 2024, so just this year, and 51,750 of those patients will die. Okay, so our overall survival rate for pancreatic cancer is very low. While this is all very bad news, we obviously want you to look at the positive side. We are making advances in pancreatic cancer. I think this is quite important to point out because if you really look at this type of cancer, although it is a rare cancer, although there's not a lot of funding for pancreatic cancer, of all the types of cancers, this is probably one of the cancer entities where we've made most advancements at understanding pancreatic cancer at the biological level. As far as genetically engineered mice, probably pancreatic cancer has the most genetically engineered mice to study this type of cancer. Tumor models, PDXs, PDOs, etc. A lot of investment has been made in trying to understand the biology of pancreatic cancer. Unfortunately, that has not translated to the patient yet, but we hope we will make advancements. And where we're making advancements is understanding a little bit better the pathology of this tumor, the omics of this tumor, at the mutational level, at the transcriptional level, at the proteomic level. We are experimenting with drug therapies, personalized medicine, trying to attack specific pathways that are upregulated in pancreatic cancer. Metabolism plays a very important role in this cancer, both glycolysis and OXPHOS. So that is also a potential Achilles heel. And of course, precision medicine. Can we take all of this information and instead of treating all pancreatic cancer patients the same, can we individualize treatments? So in my laboratory, in my group, we work on in each one of these aspects. And what I want to show you today are snapshots of accomplishments we've made, which we think are important and have helped in some way our understanding of pancreatic cancer. So let's start with pathology. Okay, so very much like breast cancer and bladder cancer, subtypes also exist in pancreatic cancer. And this is something we did not know until very recently. You can see probably in the last 10 years, we realized that subtypes do exist. And this is somewhat important because now we can classify patients based on their subtype and in the future possibly use a personalized medicine approach for these patients. To date, this is not used in the clinic. So we are not giving patients a specific type of treatment based on their subtype. But what we know based on studies from Bailey, Moffitt, and Collison is that we have a classical subtype, which is also referred to as the epithelial subtype or the progenitor subtype. And we also have a basal subtype, which is also known as the quasi-mesenchymal or the squamous subtype, which again, basically the difference between these two, this will be a very differentiated tumor and this will be a less differentiated tumor. Okay, so subtypes exist. So that means that this tumor is not as heterogeneous as we thought. There's also inter and intratumoral heterogeneity. And there's a lot of heterogeneity that exists within the pancreatic tumor. So in the laboratory, we've been trying to decide or trying to figure out how this heterogeneity is maintained and where does it come from. And we believe that the cancer stem cell model is a very nice model to explain tumor heterogeneity. So for those of you who are not familiar with the cancer stem cell model, you can see the tumor here. Cancer stem cells represent a very rare small subpopulation of cells within the tumor. These cells sit at the apex of tumor hierarchy. These cells can undergo both asymmetric and symmetric division. Undergoing asymmetric division gives rise to all of the other bulk tumor cells that we find present within the tumor, and symmetric division and via self-renewal always giving rise or never losing the cancer stem cell pool. Of course, this is a very bidirectional model that we see here, but we know that this is actually a unidirectional model, but we know this is actually bidirectional. Differentiated cells can also dedifferentiate and replenish the cancer stem cell pool if necessary. But here, simplified, you can see the cancer stem cell is a cell that's most pluripotent and the most chemoresistant, while the cells down here more differentiated, or the cells obviously that are more differentiated, and there's a higher degree of heterogeneity at this level. So the cancer stem cell model is actually not a new model. If we look back around 1855 to 1975, there were multiple studies that reported stem-like cells in different types of cancers, but we didn't understand or know that cancer stem cells existed here. What actually pathologists and physicians pointed out is the resemblance of tumor to embryonic tissue. So this is sort of the first indication that cancer stem cells may exist. But it really wasn't until 1994 when John Dick and colleagues isolated for the first time cancer stem cells from acute myeloid leukemia. And this took a long time because we really needed instruments and resources to be able to identify cancer stem cells in human tumors. And for that, we needed FACS, which is fluorescence activated cell sorting, to be able to sort specific cell populations from a tumor, and immunocompromised mice, obviously, to inject these cells and evaluate their tumorigenic potential without any rejection. So since 1994, we've discovered cancer stem cells in just about all solid tumors, including pancreatic cancer. In 2007, both by Lee et al. And Herman et al. And then subsequently, as always, in, for example, pancreatic neuroendocrine tumors in 2013 to 2015. So I think the question we need to ask is, so we know a lot about pancreatic cancer, we know a lot about cancer stem cells. Where are we today, 30 years later? So cancer stem cells have been identified in just about all solid tumors, but a lot of questions still remain. Where do cancer stem cells come from? I'm often asked, should we even study cancer stem cells? What is their exact role in tumorigenesis and chemoresistance and metastasis and relapse? If we eliminate them, can we cure cancer? And this is probably the main question we ask in the laboratory and the primary focus of our interest, or the primary reason why we're interested in cancer stem cells. So how do we study cancer stem cells? I think this is a very important question because a lot of us use established cell lines And I would be very resistant to study cancer stem cells in established cell lines. We in the laboratory always draw on primary tumor material offered to us by our different collaborations with pathologists and surgeons at the different hospitals in Madrid. And we can take this tumor and we can digest it and we can look by FACS or flow cytometry and separate cells based on markers that cancer stem cells express. So we can use fluorescently labeled antibodies that recognize a specific protein or receptor that cancer stem cells can express on the cell surface, and we can separate these cells from the other cells and study them. We can also establish cultures from primary tumor material, adherent cultures, 2D cultures, and then we can take these 2D cultures and put them in spheroid cultures, basically culturing them in low attachment plates without serum. And what we cause here is the formation of these typical tumor spheres, which are enriched in cancer stem cells. And we can also take these tumors and plant them in immunocompromised mice and establish patient-derived xenografts. From this, we can generate, again, spheres or adherent cultures and then perform a FACS analysis to separate the cancer stem cells if our starting material is very low. We generally go this route. Basically because pancreatic cancer now, we don't always get the sufficient amount of material to do all of these different stages. So I want to point out here the xenograft model, the PDX model. This is a service offered by the hospital. We see here we have a Plataforma de Modelos de Biobanco, which is established here in the hospital. This is the group. And this service is offered via the biobank. We essentially take tumors. You can establish your own circuit, take these tumors and plant them in immunocompromised mice, expand them and expand them. Depends on the investigator, how far they want to go. This would be a great example if you wanted to test different drugs on a cohort of mice that have the expansion of the same tumor. We can also establish as a service patient-derived organoids or even organoids established from the PDXs. We can also expand those and freeze them down and then offer them to investigators within the institute and outside of the institute as well. Of course, we can take these PDXs, also establish the PDOs. Vice versa. This is all stored within the biobank. Both we have a collection of frozen PDX models and PDO models. And in the end, what can you do with all this? You can do preclinical trials, drug screening, genomic analyses, transcriptional profiling, proteomics, biomarker discovery. The list is long, and I'm sure you can think of many other things you could possibly do with this model. If you're interested, contact a biobank. Sonia Barba would be more than happy to give you more information. So talking about markers, how can we isolate and study cancer stem cells? There are a number of markers we've used across the past few decades to identify cancer stem cells. These are probably more specific for pancreatic cancer, EpCAM, CD133, CXCR4, intracellular markers such as ALDH1, pumps that are on the cell surface such as ABCG2. This is a list of different types of markers on different types of cancers. As you can see, there's no universal cancer stem cell marker. And this is something we actually sought out to discover many years ago. Well, probably about 2014, very talented PhD student in the laboratory when we were at the CNIO, Irene Miranda, actually discovered a new, forgive me for this is a Mac PC error, but discovered a new biomarker, which we termed autofluorescence. These are intracellular vesicles present within the cancer stem cells. You can see them here in this immunofluorescence. These are very discrete vesicles that fluoresce green. Now, obviously, you ask, why did they fluoresce green? Very good question. I'm not going to bore you with all the details of Irene's thesis, although it's quite exciting. But what she discovered is that these vesicles were actually covered or coated in ABCG2. ABCG2 is an ATP transporter. It transports and pumps things into vesicles or outside of the cell. You can see here how these autofluorescent vesicles are covered in ABCG2. So what was happening in the cancer stem cells is that these vesicles were actually concentrating some substance inside of these autofluorescent vesicles. And what we discovered through a number of analyses and a long time of troubleshooting is that what was accumulated inside of these autofluorescent vesicles was nothing more than riboflavin, vitamin B2. And at large enough concentrations or high enough concentrations, the riboflavin would actually fluoresce green. As you can see here, this is a jar with riboflavin excited with blue light or fluorescent light, and you can see the fluorescence that's emitted by riboflavin. And we've since gone and checked to see whether or not autofluorescent cancer stem cells exist, not just in pancreatic cancer, but in other types of tumors. And we've seen and analyzed to date about 145 tumors and found that cancer stem cells are obviously present in PDAC, colorectal cancer, pancreatic neuroendocrine tumors, and a number of other tumors, which I'll get to at the end of the talk. Obviously here, this is a nice video that Diego from the laboratory generated. You can see here that this is a dynamic process. Not all the cells have autofluorescent vesicles, and not all the cells will maintain their autofluorescent vesicle. Here's an autofluorescent vesicle that is formed. And I have the mouse, I will. Sí, vale, por la mitad. Sí.
Avanza.
Sí. Ahí está. You can see another autofluorescent vesicle forming here. This one will disappear, and then other autofluorescent vesicles will appear. So this is a very dynamic process. We don't understand the kinetics behind this, but they appear and disappear. In all cases, obviously, the formation of this vesicle is because of the concentration of riboflavin inside of these vesicles. So this is our list that we've been working for many years, and since the discovery of the autofluorescent vesicle, we've actually added some more markers to understand or to identify cancer stem cells, CD47, which is a don't eat me signal, P2X7R and FPR2, anthrax toxin receptor 1, and we're always looking for new markers that can help us identify and better classify cancer stem cells. We just recently published PGLERV1 as a new cancer stem cell. This was the basis of Juan Carlos Lopez Hill's thesis that he defended last Friday, very successfully. At the omics level, what do we know? Well, because we can separate cancer stem cells from non-cancer stem cells, we can perform omics studies. For example, we can look at methylation profile, and we published that the DNA of cancer stem cells is hypermethylated compared to non-cancer stem cells. We can also perform microRNA profiling studies, and we found different microRNA clusters that are either downregulated or upregulated in cancer stem cells, and these play a very important role in chemoresistance, for example. And of course, we performed RNA-seq analyses. All these studies really just allow you to generate a list of putative targets that define a cancer stem cell population and a non-cancer stem cell population. So for the last few years, we've been really working on our results from RNA-seq analysis, and we've developed a cancer stem cell signature and also a non-cancer stem cell signature. We've identified 112 genes that are overexpressed in cancer stem cells and a number of tumor models, and 101 genes that are underexpressed in cancer stem cells. And another way of looking at this is in 101 genes that are overexpressed in the non-cancer stem cell population. So this gives us two signatures that we can apply to databases. It's exactly what we've done. There are a number of pancreatic cancer databases that exist, Bailey, Collison, FIAS, Junkie, and what we'll be able to do is apply our signature to see whether or not we can find pathways that are enriched in tumors that we classify as cancer stem cell rich tumor, or tumors that we can classify as a non-cancer stem cell enriched tumor. The majority of the tumors fall in this area. The majority of tumors that we've analyzed at all of these databases have a very strong propensity to be a cancer stem cell enriched tumor. So this allows us to look at pathways that are enriched in these tumors and pathways that are enriched in these tumors. And what you can see, for example, KRAS signaling is very upregulated in the tumors that are cancer stem cell enriched. And we also see changes in metabolism. These tumors are very OXPHOS, fatty acid metabolism dependent, while the non-cancer stem cell enriched tumors primarily rely on glycolysis to meet their energy requirements. So with this information in hand, can we use this to our advantage? Can we perform specific types of drug therapy interventions to attack tumors that are, for example, OXPHOS rich or cancer stem cell rich versus tumors that are not? So if we go back to the cancer stem cell signature, one of the pathways is upregulator KRAS signaling. So Laura Ruiz from our lab gave an excellent presentation a few weeks ago on KRAS signaling. So I won't bore you with all the details. Laura did a very excellent job, but I will just say that this is not surprising, obviously, because KRAS mutations represent 90%, are present in 90% of PDAC tumors. And we know that when KRAS is activated, basically we have essentially a very strong influence on oncogenic signaling driven by KRAS signaling. Now, for the longest time, KRAS has been considered undruggable. And recently, compounds have come out that can target KRAS with specific types of mutations. For example, G12C, which only represents less than 2% of PDAC tumors. So unfortunately, we really can't use these new inhibitors for pancreatic cancer, but they are being used, for example, for lung cancer. So the idea here is, since we can't target KRAS, could we potentially, in tumors that enrich in cancer stem cells, target KRAS signaling upstream and downstream. And as Laura showed us, there are different types of inhibitors, for example, SHP2 inhibitor that inhibits pathways or that KRAS signaling upstream, and MEK inhibitors, for example, that can inhibit KRAS signaling downstream. And in collaboration with Hannah Al-Ghul, many years ago, we showed that a combination of SHP inhibition and MEK inhibition actually was very nice at, had a very strong cytostatic effect on PDAC tumor growth. However, this is a very toxic approach, a very toxic combination. So we joined forces again with him, led by Laura Ruiz in our laboratory, and instead of targeting MEK, what we decided to do is again target SHP2 and target ERK. Laura showed these data last two weeks ago, but all of these data in their together actually led to the initiation of the SHRPA trial, which is a clinical trial that was initiated in March of 2022. It closed this summer. We're still processing the results. Unfortunately, it does not look good, primarily due to the toxicity that was given or that the patients suffered with a combination of these drugs, primarily cardiotoxicity. And also we didn't see the effect that we expected on tumor growth. Metabolism. Metabolism is another pathway that we saw upregulated in cancer stem cell enriched tumors. As you remember, the cancer stem cell enriched tumors are enriched for pathways, for example, in OXPHOS and in fatty acid metabolism. And this is something we actually saw a few years ago, back in 2015, 2016, where we just divided cancer stem cells and non-cancer stem cells, performed a very simple microarray analysis and found that we had genes upregulated in the cancer stem cells that were involved in OXPHOS and mitochondrial respiration. While the genes that are upregulated in non-cancer stem cells, you can see here, were primarily involved in glycolysis, pentose phosphate pathway, etc. So this told us for the very first time, even before we did these studies, that cancer stem cells use OXPHOS, mitochondrial respiration, to meet their energy requirements. And this obviously represents a potential Achilles heel. Cancer stem cells are very different from non-cancer stem cells at their metabolic level. You can see here this electron micrograph of mitochondria, both in cancer stem cells and non-cancer stem cells. And I think you can appreciate here, these are very well-defined mitochondria with well-defined cristae, granularity. And you can see here the mitochondria in the non-cancer stem cells are swollen. We often find them also in autophagosomes being eliminated. So this indicates, just at the electron microscopic level, differences in the metabolism of these cells. So you can say that cancer stem cells have functional mitochondria. They are OXPHOS dependent, and non-cancer stem cells have dysfunctional mitochondria, which they depend more on glycolysis to meet their energy requirements. So obviously this led us to collaborate with José Luis Mascareñas. Laura also explained this in her talk 2 weeks ago. This compound IGN-111 is a ruthenium-based compound that we've been using for many years to target mitochondrial respiration or oxidative phosphorylation in cancer stem cells. This is some data from Laura. You can see here that IGN-111 is very potent, also very cytostatic, targeting PDAC growth. You can see this was the size of the tumor when we initiated these are the tumors at the end of the experiment, but they did not change in size from when we initiated the experiments, while the tumors in the control group obviously doubled or almost tripled in size. So this is a very nice reflection of how this compound was very cytostatic and inhibited tumor growth. The nice thing about IGN-111, it's not toxic. We don't see the same toxic effects that, for example, what we saw when we tried to inhibit tumor growth with the SHP2 inhibitor and the MEK or the ERK inhibitor. So we're continuing to explore this compound. We've received financing from La Caixa. We'll also apply for an EIC transition to hopefully take this compound to the market and obviously into phase 1 clinical trials. So precision medicine. So I think this is very important. So a lot of what we've learned here enables us to really take a step back and determine whether or not a specific tumor should be treated a specific way. Okay. And so, for example, can we treat tumors that are enriched in cancer stem cells in a specific manner? And one way we can see whether or not a tumor is enriched in cancer stem cells, obviously performing RNA-seq, well, we can just take that tumor, digest it, and see whether or not it has an increased percentage of autofluorescent cancer stem cells. And this is what we've done. We've done this we started this study in about 2015. It took a very long time to analyze 145 tumors. The tumors were digested in the laboratory. They were run through the flow cytometer in the presence of riboflavin to increase our capacity to detect these autofluorescent cancer stem cells. And you can see here just a cytometry plot. Here is how we detect the cancer stem cells. So we just recently published this in October in Cancer Research Communications. Again, we analyzed 145 tumors, primarily in collaboration with the Hospital Universitario en Santa Sofía with Miriam López Gómez y Enrique Casado y Gonzalo. And here's just a sort of a dot plot or histogram of all the tumors that we analyzed, the majority of the tumors that we were able to analyze. And you can see here, for example, rectum, we find a lot of tumors, not all of them, but a number of tumors that have enriched or a very high percentage of autofluorescent cancer stem cells. And also in colorectal cancer, we found a lot of tumors that had a high percentage of cancer stem cells. Now, the goal of this study when we initiated was just to see if we could see autofluorescent cancer stem cells in other tumors. But we realized when we completed the study that we had 75 patients in the colorectal cancer group. So we asked, can we do something with these 75 patients? Can we make some clinical correlations? Indeed, we were able to. So we had 75 patients. We were able to divide these 75 patients into two groups, patients that had a very high percentage of autofluorescent cancer stem cells and patients that had a low percentage of autofluorescent cancer stem cells with all their clinical and oncological parameters. And what we were able to do is see, well, is there a difference in accumulated incidence or is there a difference in incidence rate when patients have high autofluorescence? So if you look at accumulated incidence, this tells us the total relapse over the entire time of the period. So these are resectable colorectal tumors. Usually 25% of patients relapse in 5 years. And what our study showed us with those 75 patients, we had a relapse of 25.7% of those patients relapsed in 5 years. So the cohort was a good cohort. If we looked at the accumulated incidence, if autofluorescence was high versus low, we did see a difference, but it wasn't dramatic, 30 versus 24%. The presence of positive lymph nodes is what usually determines or can signal whether or not a patient might have relapsed in 5 years. You can see here, if you just look at positive lymph nodes versus negative lymph nodes, the accumulated incidence is 37.14% versus 11.4%. So this is probably a good biomarker. So we thought, could we combine these two biomarkers to see whether or not we improve our capacity to detect those patients that might have a relapse in 5 years? And if you see here, autofluorescent high versus low in the lymph node positive patients, we see here a very nice separation. Incidence rate, again, tells us the rate of relapse. In the overall population, it was 6.9 relapses per 1,000 patients. Again, when we combine autofluorescence and lymph node positive patients, we go from 6 to 8.6 versus 9. Again, a very good combination biomarker approach. And if you look at probability of recurrence, so these are the patients here in green that have low autofluorescence and negative lymph nodes. Their probability of recurrence is very low. Those with high autofluorescence and negative lymph nodes, a bit more of a probability of survival. Obviously, lymph node positives, we can see here, but when we combine both is really where we see a higher probability of recurrence with a very good hazard ratio. So autofluorescence, we think, can be used to stratify patients. We can take tumors from patients, digest them, look at the autofluorescent positive population, and classify a patient as a cancer stem cell high or a cancer stem cell low tumor. And colorectal cancer, we've been able to do that. We want to extend this obviously to lung cancer, where we've also detected autofluorescent cancer cells. Pancreatic cancer is a little bit more difficult, but other tumors that we also believe might be amenable to study the cancer stem cell populations. So again, going back to this autofluorescent vesicle. So since riboflavin accumulates efficiently in these autofluorescent vesicles, we asked whether we could hijack this property, take advantage of the fact that cancer stem cells accumulate something that we know that they accumulate within these vesicles, and insert something inhibitory, something like a Trojan horse into these vesicles as a means of specifically targeting them. You can imagine riboflavin linked to chemotherapy, then the cancer stem cells would accumulate that inside of these autofluorescent vesicles. It's a bit more complicated. You have to do some intricate conjugating anything into riboflavin has proven very challenging. So we thought, could we do something else? So we looked in the literature. This is a study that a former postdoc in our laboratory, Adrian Valencia, initiated a few years ago. We wanted to see, well, instead of riboflavin, are there competitive inhibitors of riboflavin? Obviously, there are. So competitive inhibitors, here's riboflavin, we can see here. Here are three competitive inhibitors of riboflavin. And then we also have cofactor derivatives of riboflavin, FMN and FAAD. And in this particular study, published in PLOS Neglected Tropical Diseases, what they used is this trypanosoma model, which lacks biosynthetic riboflavin pathway. So these parasites cannot produce riboflavin, they need to take it in, and they transport it in via these two transporters. So a very simple experiment, what they saw is that if they incubated these parasites with riboflavin at different concentrations, obviously nothing happened at the level of their proliferation. If they incubated these parasites with both of the cofactor derivatives of riboflavin, FMN or FAD, saw no difference. But when they incubated these parasites with the competitive inhibitors of riboflavin, rosioflavin, lumeoflavin, or lumicrome, what they saw is a dose-dependent decrease in their proliferation. Okay, so now we have riboflavin competitors, which can be used potentially to target cancer stem cells. So that's exactly what we did. We looked at lumicrome. There was only one publication in the literature showing lumicrome inhibits human lung cancer cell growth. So we thought, okay, great, this is probably an interesting avenue of research. And what we did is incubated our cells with different concentrations of lumicrome. This is very preliminary data. It's data that we've accumulated over the last year, which we hope to continue with. What we saw here is that in the presence of lumicrome, both 1 micromolar and 5 micromolar lumicrome, we saw a very nice decrease in the autofluorescent positive cancer stem cell population in the cultures. Unfortunately, what we saw is that a lot of toxicity with lumicrome just added to the culture, especially at 5 micromolar. So this is targeting all the cells. We had a lot of cytotoxicity, which is not what we wanted. We wanted something specifically to enter in the cancer stem cells, kill just the cancer stem cells and not the other cells. So this is always a 24-hour treatment. So we thought, well, if we go a little bit less, maybe a 4-hour treatment is sufficient to reduce toxicity, but also maintain the inhibition we see on the autofluorescent cancer stem cells. So here, just a 4-hour incubation with 5 micromolar lumicrome. We have about a 50% reduction in the autofluorescent cells. And here we got less cytotoxicity. But we were not happy with this. We wanted this, but with this level of cytotoxicity. So we teamed up with Maria de la Fuente in Santiago de Compostela, which is an expert in nanoparticle development and nanoemulsions. And we thought, well, could we possibly encapsulate lumicrome or more efficiently send it to cancer stem cells? And what we saw with the lumicrome encapsulated in the nanoparticles is a very nice inhibition in the autofluorescent cancer stem cells and essentially no cancer, no cell death. And since lumicrome is an inhibitor or a competitive inhibitor of riboflavin, riboflavin participates in OXPHOS, mitochondrial respiration. What we saw also with lumicrome and also with rosoflavin is that we saw a decrease in oxygen consumption. So this is just a seahorse analysis. The green line is the control, and the lines that are below that are those that are treated. And if they're below the green line, essentially that means that they have less oxygen consumption. We are targeting OXPHOS in these cells. So that's great because we know that OXPHOS is very important for cancer stem cells. So we want to take this to an in vivo model. We've only done one experiment. It's a very preliminary data, but I think it's encouraging. What we performed is an orthotopic implantation of tumor cells in immunocompromised mice with human PDAC cells, allow those tumors to form. And then for 3 weeks, what we did is we treated these mice with the nanoparticles that contained lumicrome and plus or minus gemcitabine to see whether or not there was a synergistic effect. So here are the results. What we do see is that with the lumicrome nanoparticles alone, we see about 50% reduction in tumor weight and also metastasis weight, so overall tumor burden. And also with gemcitabine and lumicrome plus gemcitabine, we also saw a more effective, more dramatic effect. Now, what we want to say, okay, this doesn't seem that the lumicrome and gemcitabine together are synergistic or additive. We do know that gemcitabine alone, when you give it to, in these orthotopic models, if you remove gemcitabine, relapse is very fast. Gemcitabine attacks the more proliferating cells, does not target the cancer stem cells. The cancer stem cells survive after gemcitabine treatment, and we see relapse very quickly. So we wanted to see in these tumors whether or not there was an enhancement or enrichment in cancer stem cells. And you can see here, both looking at autofluorescent cancer stem cells and CD133 positive cancer stem cells, we've seen the gemcitabine group compared to the control groups, and increased enrichment in cancer stem cells. So again, holding true to what we know, gemcitabine treatment does reduce tumor growth, but does enrich for cancer stem cells. And what we saw with Lumicrome, we did not, with the combination effect, gemcitabine plus Lumicrome alone obviously did not enrich for cancer stem cells. It was actually targeting cancer stem cells. And we see that together, we actually inhibit this effect that we see with gemcitabine alone. So there could be a potential of combining these two treatments. And also at the level of metastasis, this is your tumor cells that we found in the liver. We also see that all three of the treatments, including the Lumicrome nanoparticles by themselves, were very effective at reducing the capacity of these cells to make it to the liver. So we do feel that we can take advantage of the fact that cancer stem cells concentrate riboflavin in these vesicles, perhaps put something else inhibitory in, use a basically a Trojan horse approach, and obviously send something to these vesicles that can inhibit cancer stem cell proliferation. So we were happy with the Lumicrome effects, but we thought we could be a little bit better. And we know, for example, that one of the ways to sterilize blood, for example, is to add riboflavin. These blood or these samples are passed through broad spectrum UV light. Usually, if you're unfamiliar with, if you want to eliminate bacteria, for example, bacteria love to take up riboflavin. And if the bacteria take up riboflavin or are present, for example, in a biological fluid or blood, and you pass ultraviolet light, that light will act upon the riboflavin, induce Ross oxidative species, and will kill the bacteria. So this is one way, very nice way to clean blood, for example, that's used, for example, transfusions. So we actually thought we could possibly induce light within the tumors, and that could increase the capacity of Lumicrome to inhibit the cancer stem cells. Obviously, for that, we have to, in some way, get light into the tumors. And this is what we're working on right now with Maria de la Fuente. And what we've been able to do is develop a bioluminescence photodynamic therapy approach with another type of nanoparticle that she's developed. So this is the control mouse. You can see the tumor here on the right. This is an orthotopic tumor. This is the sham. Basically, it gets all of the injections, but doesn't get the substrate that induces the light. And this is the mouse where the substrate has been added to induce light within the tumor. And we can see very nice with IRYCIS that we are able to send nanoparticles to the tumor, and we are able in these tumors to induce light. Now, we'll combine this technology with the Lumicrome nanoparticles to see whether or not light can enhance even more the capacity of Lumicrome to act on the cancer stem cells. Again, all very preliminary data, but this is an area, a line of research that we are continuing to study in the laboratory. And so I promised the Fundación that I would be in 40 minutes, which I think we're on time because we want all of you all to go to the Jornada de los Postdoctorales, which I think will be probably more interesting than my talk, but I will end this just obviously thanking my group. They're really the ones that do all the research on this here, selling what we do and trying to sell in the best way possible. Obviously, our cancer stem cells and fibroinflammatory and microenvironment group, the BioPack group, this is a picture of all of us together. And of course, we can't do this without all of our collaborators, Maria de la Fuente, Hannah Albul, José Luis Mascareños at the IGM 111, Laura García Barmejo here in IRYCIS, Miriam at Hospital Universidad de Infanta Sofía, which helped us, which was the key motivator of the cancer stem cell autofluorescent study in colorectal cancer. And of course, our funding agencies. And with that, thank you very much. I'll be happy to take any questions. You can ask the questions in Spanish, in English, however you want. Thank you.